Hey, Jut.
Hey, Joe. Hi, everybody.
Good to see you, my friend.
Always.
All right. Well, we were rolling right along. We were just in the deep sea. Now we're going to go deep into caves with you. I think it's going to be a lot of fun. We're going to get lost for a little bit.
All
So,
right.
all right. I want to give you a quick little introduction, then I'll let you take over. So, joining us right now at the Global Biodiversity Festival, we have Jut Wynne. He's bushwhacked through the jungles of Belize in China, rappelled into the deepest volcanic pit on the big island of Hawaii, endured the world's driest desert in northern Chile, explored the deep vertical caves in southern Spain, and he does this all to learn more about caves and the secrets they may contain. He's even gone to one of the most remote inhabited places on the planet, Easter Island. And I do want to add this little caveat because this is super cool. He's also very active in developing planetary cave exploration, working with NASA researchers to find ways of using robots to examine and explore caves on other planets. So absolutely amazing. So great to have you joining us live today, Jut, and we are excited for the biodiversity in caves.
All right. Once again, very glad to be here. And without further ado, we will get this underway. All right. So what I'm going to be speaking about today is we're going to be going deep. Specifically, we're going to be looking at cave biological diversity. And in so doing, there are several things that we should be thinking about here. And first off is, okay, how do things evolve in general? So when we think about that, there are two scales in which evolution can occur. First of which is microevolution. Now, these are small changes which can happen individually, and actually as the environment changes, what can happen is it can affect the survivability of different different individuals within a population. I like to equate this to trimming the sails when you're sailing. So when you're sailing, you trim the sail to where you maximize the efficiency of the sail and thus your speed. So that's basically what microevolution is doing. It's making these micro changes to the population to make it better fit to the environment. And then when we look at macroevolution, obviously this is at the much larger scale. And what we see here is we see these speciation events occur. And this typically happens within the context of what we call genetic drift. And what genetic drift is basically it is driven largely by isolation, and what happens and it's typically observed in much smaller populations where you see these changes in the genetic structure of a particular population, and over time what can happen is that can result in a new species developing, and that's called allopatry via isolation. So a population inhabits a new area, it becomes isolated from the parent population up here, the new population is here, and there is no interaction between the two populations, and allopatry occurs, and thus a speciation event results. And this is something else that's very important to remember, is that the environment selects. We think that animals are fitting into, that they're selecting the environment, but it's actually the other way around. It's the pressures that the environment places on a population of animals or plants, and that plant or animal species it can either fit within the environment and evolves to fit within the environment, or it doesn't. And when that happens, an extinction event could occur. So what is a species? Now, this is a really cool question as well, because this is something that's been hotly contested with evolutionary biologists and ecologists for many decades now. But the definition that I'm going to use for this particular talk is the biological species concept, which was first proposed by Ernst Mayr in 1996. Basically, what it says is that a successfully interbreeding population that can successfully interbreed from one generation to the next. And if that occurs, then you have yourself a species. Something else to consider is this notion of adaptive radiation. And perhaps one of the best examples of this, and the first example actually, was with Charles Darwin and his observations on the Galapagos Islands. Now, these are Galapagos finches, also known as Darwin's finches. And what happened was a founder species arrives on the islands, and it was the first to be able to, that was successful. And as a result, it was able to radiate out into these niches. And these are habitation spaces, or habitat spaces, which are not occupied. And when that happens, you see these speciation events. And one of the telltale signs here that you can key in on when you look at these finches and when you look at the next entrance, slide rather, just look at the differences in beak shape. And that is one of the things that Darwin really keyed in on when he made this observation. And he published his book on the origin of species in 1859. Here's another example. I am very fond of the Hawaiian Islands, do a lot of work there. Hopefully we'll be doing a lot more work there. And I've learned a lot about the honey creepers. This is another type of animal that underwent adaptive radiation. There was a founder species that arrived, and it quickly radiated and into all these new niches, resulting in this rapid divergence or speciation process, which resulted in, you can see here, up to 27 different species of honey creepers once occurred on the Hawaiian Islands. So now let's hone in on cave evolutionary biology. So it's very important when we think about the evolution of cave organisms to recognize that caves are zonal environments. And we have 4 primary zones here: light, twilight, transition, and deep. And what you have in terms of the climate of the caves is you have low climatic stability at the entrance, gradating to the highest stability at the deepest part of the cave. Now, as you would expect in the entrance of the cave, what we have is, well, the rising and the setting of the sun results in temperature fluctuations, which are also felt within the cave. And then, of course, seasonality. So whatever happens on the surface is largely felt within the entrance of the cave. However, if you go to the deepest part of the cave, that is the most stable part of the cave. And if deep zone conditions do exist, what you have is you have a near stable temperature, which reflects the average annual surface temperature. It has a near water saturated atmosphere. And one of the ways you can typically, if you've been in enough caves, you can smell it. You can tell when you're entering the deep zone. But in other ways, if you exhale, you can see your breath. And that tells you that you're in the deep zone. Other aspects of the deep zone, little to no airflow. And as you would expect, it's very deep, so it's going to be completely dark. It's aphotic. And as one would expect, in terms of speciation or adaptation to the cave environment, we're going to see low specialization in the entrance, and we're going to see very high specialization in the deepest parts of the cave. And by that, I mean animals that are adapted, they're tightly linked to the cave environment, and they're adapted to the cave ecosystem. Now, for this talk, what I'm going to do is I'm going to provide you with bookends, the 2 different types of cave-dwelling animals that we typically encounter in caves, and these are the 2 endpoints, right? So a troglophile is an animal. It's not cave-adapted. It can complete its life cycle underground. It's perfectly happy doing so, but it can also be found in similar surface or cave-like surface environments. So, for example, beneath a log or underneath a rock, where you'll have these damp and dark environments or habitats for these animals. The other end of the spectrum are subterranean-adapted animals. The first of which is called a troglobiont. And now this is an example of, this is actually, wow, this is a classic example of a subterranean-adapted animal. And you can see here it's depigmented. It's this yellowish, whitish color. It has these long, slender legs. It has long antenna. It's eyeless. And and you can see it's kind of difficult in this image, but you can see it's covered in hair or setae. So it's very common that these animals are very hairy. And these long antennae and the hair all over their bodies become their eyes in complete darkness. Now, a stoglobiont is simply the counterpart to the terrestrial subterranean adapted animal. It's basically the aquatic version of a troglobiont. So how do animals ultimately become troglobionts or stagobionts? How do they become subterranean adapted? Now, this is a schematic that Tom Barr proposed back in 1968, one of the earliest folks to be thinking about these types, this idea and concept. And so you can see here from the title of the slide, there are these three things that typically occur. The animal colonizes the cave. Over time, it becomes isolated from the surface population, and then it may ultimately evolve into a subterranean adapted form. And you can see here what we have is what's called E1, and this is just for surface dwelling or epigean species. It will, the surface dwelling population will colonize a cave, and it happens multiple times here. And as a result, you have multiple different species that ultimately evolve from this one surface dwelling species. And the idea being here is with this circle with the line slash through it is indicative of that population ultimately goes extinct. And the subterranean populations evolve into new species and they remain. Now, this is something that Frank Howarth proposed back in 1973. He was working in lava tube caves on the Hawaiian Islands, and he noticed that they were, highly fractured and cracks and crevices. And he posited, wow, I wonder if animals can use those cracks and crevices as highways, as a means of getting through to other caves, perhaps on a larger landscape scale. So what he proposed is that a surface dwelling population colonizes a cave, and then the population is quite happy down here. The surface dwelling sister population either goes extinct or it doesn't. And then what happens is over evolutionary time, you have a subterranean adapted species that evolves, and then because of these cracks and crevices, it can move freely and ultimately colonize multiple caves on the landscape, having a much larger distribution. Turning our attention to what drives biological diversity, because that's why we're all here, right? We want to understand and celebrate the importance of International Biodiversity Day, which was on Friday. So there are two principal drivers that I want to delve into in terms of what causes there to be so many species in some areas and not so many species in other areas. The first of which is called the latitudinal diversity gradient. And the best example that we can come up that I can provide you all with is comparing the number of plant and animal species in the Arctic tundra in comparison to the plant and animal species diversity in a tropical rainforest. Obviously, there's going to be a huge difference in terms of diversity. There's going to be far more species in the tropical rainforest. And the reason that is, and there's a couple of reasons, is one, during the last ice age, the more temperate parts of the planet were covered in ice, or largely covered in ice. And the tropics, the equatorial region, was not. So what you had is first you didn't have the ice coverage. And then secondly, you had longer stability in terms of the environment along the equatorial region, which ultimately gave rise to a higher level of diversity in the tropics versus the more temperate regions. Also, there's this notion of habitat heterogeneity. So we know on a landscape, you know that there are different types of habitats. So typically in nature, and as a result, you will see different numbers of species in those different habitats. I live in northern Arizona in the world's largest ponderosa pine forest, and it's relatively uniform. There's ponderosa pine, as you would expect, and there's gamble oak, and there's grasses and forbs, and so there's a certain number of species occurring within that habitat. But if you look at the canyons that finger that into the lowland areas, that those are going to have much higher diversity because they're going to be shaded. There's going to be water. There's going to be different types of tree species that can capitalize on the fact that there is shade and water. And that will give rise to higher numbers of animals and as well. So let's explore this idea of a latitudinal gradient. So we're going to look at it compare two different study areas. We applied similar techniques in terms of collecting the animals in both of these areas. First of which are Grand Canyon caves, and then we're also, and this is Grand Canyon Parashant National Monument, northwestern Arizona, on the north rim of the Grand Canyon. The other are caves in Belize at Runaway Creek Nature Reserve. You can see here there is a lot of difference in latitude between these two areas. So first, let's look. This is a pie chart for Grand Canyon Parashant. We sampled 6 different caves, and we had 13 different groups, and those groups are identified by these pie slices within the pie chart, and they were either family level or higher. So in this particular case, we had mostly orders. What we found was we had 44 different species of cave insects occurring within Grand Canyon Parashant. Of those, 7 were troglobiotes. Now let's see what we found in Belize. In Belize, Runaway Creek Nature Reserve, wow, just first off, just compare the number of pie slices from Belize to Grand Canyon. There are a lot more pie slices, right? So what that's telling us is that there are more groups, taxonomic groups, represented in Belize, and there are from Grand Canyon, 22 versus 13. We see similar patterns when we look at the number of species. 64 species were identified from just 4 caves in Belize, 16 subterranean-adapted animals. So what we're seeing here is nearly 1.5 times as many species in Belize when compared to Grand Canyon, and more than twice as many subterranean-adapted animals. And there's other things going on here. One, Grand Canyon is high desert. It's pinyon juniper woodlands and Mojave Desert, so it's going to be much drier. And of course, you have the tropics, the tropical dry forest in Belize. So you have those two differences in habitats or in environments, which is part of this this latitudinal gradient that we're seeing here. So let's look at habitat heterogeneity. So when it applies to caves, what we've observed over the years is that oftentimes in the entrances, there's sheltered environments so they can support relic plant communities. And what a relic plant community is, it's a composition of plant species which occur within the cave entrance and might not occur anywhere else on the landscape on the surface. So these are isolated communities. And because there's plants, well, oftentimes there are also relic insect populations, and i.e. species that occur within these habitats. This is an example from western New Mexico. Here's another example of relic plant communities from Easter Island in the South Pacific Ocean. And yet another example from the South China Karst in Asia. We found unique animal species or insect species specifically within these communities, and oftentimes these plant communities were unique to the surrounding landscape. Other examples of heterogeneity is nutrients. So caves are often considered nutrient-starved environments. And bats, well, they kind of solve that problem for some animals. When there's a lot of bats, as you can see in this image here, they bring nutrients from the surface into the cave in the form of guano. And what we can see is entire insect communities that are centered upon this food resource. And in some cases, folks have argued that if you remove this nutrient source, you could adversely affect those communities and potentially result in a collapse of that ecosystem. Also, the presence of dead bats. So when bats die, they fall from the ceiling, rest on the floor, and they become food for other cave-dwelling organisms. Now this is a really cool one. These are root curtains. And this is something that occurs within caves as well. And these don't occur uniformly from the entrance all the way to the back of the cave. They'll occur in little pockets within the cave, hence heterogeneity within the cave system itself. And what we see here is we see subterranean-adapted animals that have formed pretty impressive communities that are 100% reliant on the presence of these roots. You remove the roots and you could potentially drive some of those species to extinction. This is an example from Hawai'i Island. And what happens here is the roots, obviously, as you can see, they come from the surface. They work their way through the cracks and crevices and into these open subterranean spaces. Now, this is something that we're working on. So this is not quite hot off the presses, but it's in preparation. This is a paper that we're working on looking at the relationships between ohia tree, this is a native tree that occurs on the Hawaiian Islands, and its relationship to species diversity of arthropods. And this is another example of the root curtains. And you can see here that what we're looking at is we want to understand how the presence of those root curtains are affecting the diversity of the community. So if we see more root curtains, are we seeing a higher diversity, less root curtains, a lower diversity? Those are that and other questions are what we're presently looking at with this study. So let's look at an example now comparing millipede diversity between southern China and Grand Canyon. These are our study areas. This is Guangxi province here, Grand Canyon caves once again in the southwestern US. This is a nice picture of northern Guangxi province and where the caves occur up in the mountains. And this is Guangxi once again, and this is the area that we were working in. We sampled 4 caves in the Guilin area. And here are some of the animals that we encountered. This is a troglophile, another troglophilic millipede. And what I want you all to recognize here when we're looking at these images is just look at how different each one of these species of millipedes are. This is a troglobiont, subterranean-adapted animal, another subterranean-adapted animal. Here's another, and we'll end on my favorite, the dragon millipede. So you can see that there is a very impressive diversity that was encountered in just 4 caves. Now let's turn our attention to Grand Canyon. We sampled a lot more caves in Grand Canyon, and I'd also like to emphasize that we spent a lot more time. In China, we had about 2 hours per cave. In Grand Canyon, we probably spent 10+ hours per cave. We went there multiple times, applied all sorts of different sampling techniques, and what did we come up with? 1 millipede species. The difference is very impressive in my opinion. So here, let's just compare the differences here. So we're looking at Guangxi province once again, 9 species from 4 caves, spending just a little bit of time looking for them. It resulted in 6 new species. 4 of them were subterranean adapted. Looking at Grand Canyon, once again, only 1 species from 4 different caves. And one of those, you can see here, was cave restricted. So what's driving millipede diversity? All right, so first the question is it the latitudinal gradient or is it habitat heterogeneity? Well, clearly, we're looking at 2 different scales, right? So first, when we look at the landscape scale, the global scale, you can say, well, of course, it has to be the latitudinal gradient. There were 9 species in 4 caves versus 1 species that was observed in only 4 caves when 17 were visited. So you see the latitudinal gradient at play, but when you look at habitat heterogeneity, well, what do millipedes like? Well, they like damp, dank areas. They like areas with high humidity. You find them typically under rocks. Here in Arizona, you see them, these very large millipedes will come out after the monsoonal rains. In the caves on North Rim, there were only 4 that had really pronounced deep zone conditions. And so there was a lot of water. There was high humidity in the caves. There was actually standing water in pools in some of the caves. And those caves periodically flooded, bringing in vegetation from the surface. And when that happens, that provided a food resource for the millipedes. So at the cave scale, we were seeing habitat heterogeneity at play. So it can be quite complicated in terms of what the drivers are. So to wrap up here, what I wanted to emphasize is that I hope that this helped to improve your understanding of what some of the drivers are for biodiversity, and how biodiversity is indeed a global phenomenon, and it varies from place to place. Also, it's important to emphasize that there were similar patterns that have been observed on the surface for quite some time that are also being observed underground, principally the latitudinal gradient and habitat heterogeneity. And finally, what I hope is that this inspires you all, that when you're walking through a forest or when you're walking through a field, that you stop and wonder what biodiversity may exist right beneath your feet.
All right, Jut.
And I would like to thank you all.
Oh, sorry, I jumped in too soon. Keep going, Jut.
Oh, no worries. I just wanted to thank everyone so much for taking the time to join me on this Sunday. And I'm happy to take any questions at this time now.
Excellent. Well, let's get you back from that screen share, Jut. We'll take a question or two, and then we're going back to the ocean after this.
All right.
All right. So first of all, we do hear a lot about the ocean and the new species to be found, but you've definitely introduced us to an environment that needs a lot more exploration, because there's a lot more to find if you go underground. So that's really cool, Jut. So where would, in an ideal time where we could travel more freely, where would your next place be? Where would you want to go and find? It looks like incredible finds in China. Would you head back there, or is there another place on your list?
Oh, goodness. A lot of different places. Right now, Belize might be coming into focus in September, provided, we get on the other side of the pandemic. But yeah, right now, the big kibosh has been placed on just about everything in terms of travel and field work. It's very difficult. Obviously, caves are confined spaces, so there are concerns about us being essentially on top of one another underground. So there are a lot of things that we have to contend with. Also, something that the U.S. Fish and Wildlife Service is thinking about here in this country is whether or not humans can give COVID-19 to bats, and then bats ultimately giving it back to humans. So that's something that has also put somewhat of a damper on our abilities to go back underground anytime soon. So we're kind of waiting for directives from the Fish and Wildlife Service in terms of what our next steps are going to be. But yeah, there are so many places. China's definitely one of them. But right now, I guess I, as most people on the planet, are in a holding pattern at the moment.
Yeah. Well, Jut, thank you so much for representing the caves. You're the first one in 3 days to come in and talk about caves and cave biodiversity. It's a whole new world under there, and I hope you have people thinking in a different way about what's beneath our feet. We're going to jump into the ocean next. We have Sylvia Earle coming to join us. But again, I want to say, Jut, thank you so much for joining us live today, Arizona, and I hope you can get back underground soon.
Excellent. And I got to open up for Sylvia Earle. Hi, Sylvia.
All right.
I warmed it up. I warmed up the crowd for her.
Perfect. Perfect. Well, Jut, we'll talk soon, and we'll follow your next adventure. Looking forward to it.
Joe, thanks again. Always a pleasure, man. Take care.
All right.